So remember last time we were talking about how to grow viruses. Remember we said that viruses need a host, right, to grow in. So with bacterial viruses, we grow them with bacteria. We spread that mixture on the surface of agar plates, and the bacteria grow and cover the entire surface with growth. And then the viruses reproduce and kill the bacteria, leaving holes in that growth. And we call those holes plaques. And of course, we will count them, and we'll determine the PFUs per milliliter, or plaque forming units. And what forms a plaque is a single virus particle. And then we said for animal viruses, it's a bit more complicated because you have to grow them in an animal host. And a common host is a fertilized chicken egg, because that fertilized chicken egg has a developing embryo in it. It's undergoing rapid mitosis and cell division, very high metabolism, so that's what the virus wants. And we also said we can grow viruses in tissue culture. And so when you do that, you can observe the effect that viruses may have on those cells. And so when it comes to animal tissue, when you grow those tissues in culture, the cells spread out and form a monolayer, a single layer of cells. And that's because animal cells have signals that tell them to stop dividing. One of those signals is contact inhibition, or density-dependent inhibition. So the idea is when the cells are touching each other on all sides, they stop growing, they stop dividing. Cells also typically show anchorage dependence. So the idea that the cell must be anchored, it must be attached to a substrate in order to divide in the first place. And so when you infect cells with certain viruses, they can change how cells behave. And one of the changes is that cells can lose contact inhibition. And so that means the cells just keep on growing and dividing and dividing, and they grow on top of each other, forming masses of cells called a tumor. Now, not all tumors are cancer. Some tumors are benign, like warts. Others are cancerous, they're malignant, and they spread. And so cells also may lose anchorage dependence. So that means they don't have to be attached to a substrate to continue to divide. And that's what happens with a lot of malignancies, is that the cancer cells will break off and float away, and they'll start dividing and forming tumors elsewhere in your body, but they can do that without being attached to a substrate. And so we can observe how these cells get transformed by certain viruses, and that can help to identify certain kinds of viruses. When it comes to identifying viruses, one of the things we look at are those cytopathic effects, right? Changes in the host cells. And so here, in these images on the left, we see normal, healthy cells which have formed a monolayer. On the right, these cells have been infected with a virus and they've been transformed. You notice that a lot of the cells have turned round and they're growing on top of each other, so they lost contact inhibition and anchorage dependence. And so these effects can help us to identify certain viruses. But we still have to perform more sophisticated serological tests, like using antibodies. Remember when we talked about ELISA, the enzyme-linked immunosorbent assay? The idea you had those plastic wells where you could put the antigen in and then take serum from the patient, and if they have antibodies, the antibodies will stick to the antigen, and then we can detect those antibodies. Well, so we can do those kinds of tests, and they are done with many different kinds of things like HIV, or now COVID-19. We can use antibodies in neutralization tests, viral hemagglutination, or Western blots. And remember, Western blots are gel electrophoresis using proteins, so we separate out the proteins, and then use a labeled antibody to see if a particular protein is present. So we we generally have to use more sophisticated testing to identify viruses as compared to bacteria. When it comes to nucleic acids, remember we can run agarose gels, and so we can cut up the viral DNA using restriction enzymes, and then we can compare the banding pattern to known viruses and see if it matches. So we've already talked about that kind of thing. The fancy name for the banding pattern is RFLP, which stands for restriction that's ION, fragment length polymorphisms. So RFLPs is the banding pattern, so we can see that in the gel. And remember, if you don't have enough of the DNA or the RNA of the virus to run a gel, you can make more using PCR. We all remember the polymerase chain reaction, right? So now we're going to focus specifically on bacterial viruses or bacteriophages. And so when it comes to virus replication, when a virus makes more copies of itself, all viruses follow the same 5 steps, the same 5 stages, with some modifications. So as we'll see, attachment, penetration, biosynthesis, maturation, and release. All viruses follow these same steps. It's just there may be some differences at certain steps. But the first thing any virus must do is attach to its host cell. And all viruses use cell surface receptors. So all cells have different kinds of receptors on their surface. They serve a variety of functions. And viruses have evolved to exploit some of those receptors and use them for attachment. And that's what allows for the specificity of the infection. Remember, viruses do tend to be host specific. And within that host, they tend to be cell specific. And it's because of the types of receptors on those cells. Certain viruses have evolved to only attach to certain receptors and not any others. So when a bacteriophage lands on a bacterial cell, it uses its tail fibers to attach to the cell surface receptors, and then it actually uses lysozyme, an enzyme that breaks down cell walls of bacteria, so that it can inject its DNA inside the bacterial cell. So the sheath contracts kind of like a syringe and squirts the DNA inside. So the capsid, the sheath, the tail fibers, all the structure of that virus stays on the outside of the bacterial cell. Its only true purpose was as a DNA delivery system, simply transferring DNA from one host cell to another. So only the DNA gets inside of the bacterial host. Once inside, the viral DNA takes over. Usually the bacterial chromosome gets degraded, so the only DNA the bacterial cell has to work with is viral DNA. And the bacterial cell replicates the viral DNA. It transcribes and translates viral proteins. And then all those proteins come together and assemble to form new viruses. And packaged inside the capsid is a piece of the viral DNA. And then those viruses will use lysozyme again to completely break down the cell wall and rupture that host cell so that the viruses can float away and infect other cells. So because the end result for the host cell is death, is lysis, this is referred to as the lytic life cycle of bacteriophages. So the end result for that bacterial host cell will be death. Now, as many as 200 new virus particles can be produced and released from each host cell. And the fancy word for virus particle is virion. So virion just means a virus particle. So up to 200 new virions get produced from a single bacterial cell.
Does this happen only in bacteria, not in humans? In like animal cells? The lytic cycle?
Here we're just talking about bacterial viruses specifically. When we get into the animal viruses, we'll see that they do destroy animal cells as well.
Oh, okay.
So here we see a typical bacteriophage. Remember, it's a complex virus because it has all these additional structures, the sheath and the tail fibers, in addition to the capsid. So it's kind of like a little alien spaceship landing on the bacterial cells. It uses the tail fibers to attach to cell surface receptors, uses the enzyme lysozyme in order to drill a hole through the cell wall so the sheath can contract and squirt the DNA inside. Now that bacterial cell has been infected. Notice that the capsid, all the rest of the structures do remain on the outside of the bacterial cell. Once inside, viral DNA takes over. It forces the bacterial cell to replicate, transcribe, and translate to make more copies of the viral DNA as well as viral proteins. Everything assembles. Viral DNA gets packaged into the capsid, and the viruses use lysozyme again to completely rupture the host cell wall and float away, where the viruses just look for new hosts to infect. So we'll watch a video comparing a lytic cycle with the lysogenic cycle that we'll talk about next. But are there any questions about the lytic life cycle? So in lysogeny, the virus, when it infects the bacterial cell, will go dormant. And so the lysogenic life cycle allows the bacterial host cell to survive. And what actually happens is the viral DNA gets incorporated into the bacterial chromosome. And we call the incorporated viral DNA a prophage. So in this case, the viral DNA will recombine with the bacterial chromosome and insert itself and become a part of that bacterial chromosome. So that bacterial cell is alive and well, at least for the time being. But every time that bacterial cell divides, each daughter cell has a copy of the prophage. So in this way, the virus can potentially reproduce in far greater numbers because, as you recall, a single bacterial cell can form billions of bacterial cells in a day under ideal conditions. So if this were a bacterial cell with a prophage, those billions of new bacteria all have a prophage. And so all those bacteria are infected. So the typical phage that we talk about for lysogeny is this one here. And what do we call that Greek symbol? Who knows the Greek alphabet? What's that symbol? What's the symbol here? Nobody knows their Greek alphabet?
Is it
Nobody
like
knows.
lambda or
It's
something?
lambda, right? It's lambda. So lambda phage is the textbook example of a lysogenic phage. And so when lambda infects, it does produce some repressor proteins, which keeps the prophage dormant, so it's not transcribed. And the bacterial cell just goes on its normal activities. It lives, it grows, it reproduces, it divides, and makes more bacterial cells. And as I said, each cell is infected with a dormant prophage. Now, at some point, though, that prophage can decide to leave. It can decide to excise from the host chromosome. So excise means to cut it out. If lambda excises, if it cuts itself out, it will then follow a lytic life cycle. More copies of the virus will be produced, and they will kill that host cell. But at least for the time being, that host cell is perfectly fine. It's just carrying a dormant lambda phage. Now, what can cause a lambda prophage to cut itself out? It could be a random chance spontaneous event. It could be caused by UV light. Maybe all those thymine dimers, cause the lambda prophage to say, I need to get out of here, right before this cell dies from all those thymine dimers. Or certain chemicals might cause it to happen, or in the case of lambda, it's actually temperature dependent. So lambda is what we call a temperate phage. It's dormant at one temperature, so it's lysogenic at one temperature and lytic at another. But it is the classic example of a lysogenic phage. Now, here is the cycle. Lambda does not have tail fibers, but it still attaches to cell surface receptors. It still uses lysozyme to drill through the cell wall and inject its DNA. The DNA has to circularize before it can recombine and integrate with the bacterial chromosome. But if it does so, it goes dormant. And we call the integrated viral DNA a prophage. Every time that bacterial cell divides, each daughter cell has a copy of the prophage, right? So thereby potentially creating many, many more copies of the virus this way. But at some point, the prophage can leave. It can excise or cut itself out. If that happens, then lambda will follow a lytic life cycle. Its DNA will be replicated and transcribed and translated. New viruses will be produced, and they will destroy the host cell. So let's watch a video comparing the two. So I'm going to share my screen while we watch the video. This is just a nice little animation. Just not working right now.
America needs a cure for COVID-19 now, and innovative biopharmaceutical companies are rising to the challenge.
The T4 phage initiates infection of an E. coli bacterium by recognizing cell surface receptors of the host with its long tail fibers. A recognition signal is sent through the long tail fibers to the base plate. This unravels the short tail fibers that bind irreversibly to the E. coli cell surface. The base plate changes conformation and the tail sheath contracts, causing GP5 at the end of the tail tube to puncture the outer membrane of the cell. The lysosome domain of GP5 is activated and degrades the periplasmic peptidoglycan layer. The remaining part of the membrane is degraded, and DNA from the head of the phage can travel through the tail tube and enter the E. coli. After the life cycle is complete, the host cell bursts open and ejects the newly built viruses into the environment, destroying the host cell.
So that was just a nice animation. Here we'll look at more of a lecture comparing the lytic and the lysogenic cycles.
Hey guys, how you doing? This is a video to show you the difference between the lytic and lysogenic cycles of how bacteriophages can reproduce. Just remember that this process, or both these processes, have only to do with bacteriophages, which is viruses that attack bacteria. It doesn't happen in animal viruses like the flu and other things. So basically, to kind of give you a sense of what's going on here, we see that here we have a bacteria. So this is the bacteria right here, and this is the chromosome of the bacteria, and this would be a bacteriophage. This is the virus that attacks the bacteria. So if we kind of see what's going to go on here, this particular virus is going to, as you can see, inject its DNA into the actual bacteria. And so now this bacteria has its own DNA that has the DNA from the actual bacteria. And so if we see what happens from this point on, we'll now see that the host DNA has been destroyed. The base of the phage DNA, the genes of the phage DNA directed the host DNA to fall apart. And so now the only DNA that's left in the actual bacteria is the DNA from the virus. And I'm sure you can figure out what's going to happen from this point on. Now, of course, the ribosomes of the bacteria are going to be producing proteins. These proteins are viral proteins. And as you can tell here, the phage DNA is going to now code for the production of more viruses. And of course, now these viruses are going to at some point explode from the cell, and that is basically the lytic cycle. And of course, lytic means to lyse, or it means to cut or break open. So this results in the destruction of the bacteria, and that is basically the lytic cycle. So now we're going to go through the lysogenic cycle. So now this is the lysogenic cycle, and this differs in a couple ways, and I'll talk about them after. So the first part of this is the same, that basically a virus is going to attack the bacteria. And so what they didn't show here is that the virus would have landed here, it would have incorporated its DNA exactly the same as it did in the lytic cycle. But here's now the difference. In the lysogenic cycle, we see that instead of the DNA from the virus destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now the difference is that, there is no destruction yet. So the beautiful thing for the virus is that, well, basically, it's not going to be making a lot of its babies, for lack of a better word, or offspring immediately, but this cell is going to start to divide, and it's going to divide zillions of times. And every time it divides, as you can see, that little teeny piece of DNA from the phage is going to, it's basically referred to as prophage DNA, is going to be carried from generation to generation. So now you have potentially millions, if not hundreds of millions of bacteria that all have this little piece of viral DNA. So at some point, when this host cell becomes either stressed or exposes, ultraviolet light or some kind of chemicals, a system called phage induction happens. Basically what happens, you'll see here that that little piece of viral DNA pops out, and now basically the cell goes through the lytic cycle. So the second half of the lysogenic cycle is basically the lytic cycle. So what's going to happen? It's going to destroy the host DNA, it's going to start making the proteins, and there we go. So the difference between these systems is that in the lysogenic cycle, the chromosome, or the nuclear DNA material from the virus, incorporates into the actual phage DNA. And is carried generation to generation. So the bacteria doesn't make it, or the virus does not make as many offspring as quickly, but it is incorporated its DNA into many, many more cells, and thus giving the ability to potentially make much more offspring over a longer period of time. And so that is basically the difference between the lytic and lysogenic, and I hope you enjoyed it.
I don't know, did you guys enjoy it? He didn't. Yeah, he sounds like a pretty laid back guy. I don't know who he is, but. So there are consequences of lysogeny, and these are positive consequences to the bacteria, negative consequences for us. So one of the positive consequences to those infected bacterial cells is that if they're in an environment with many other lambda phages, if they're already infected with a dormant lambda prophage, they are protected from lytic lambda phages. So if they already have a dormant prophage inside them, they cannot be infected and killed by a lytic lambda phage. So it's a weird kind of mutualism where they both fit, right? Because the lambda phage has a host to live in, the host is reproducing and making more copies of the prophage, and the host is protected from being killed by lytic lambda phages. Of course, at some point that prophage, right, can wake up and excise and follow a lytic life cycle, but at least for the time being, that host cell is protected. Now, the other consequence that's kind of good for the bacterium and bad for us is kind of a weird thing. So when we look at these pathogens, Corynebacterium diphtheriae, Clostridium botulinum, Vibrio cholerae, so these are all dangerous pathogens, right? Diphtheria, botulism, cholera, so they're all very dangerous. But here's the thing. They're only pathogenic because they're infected with a lambda prophage, and that lambda prophage has brought with it the toxin gene. So the toxin for diphtheria is encoded by a lambda prophage. And the same for botulism, remember Botox, that really deadly neurotoxin, and cholera toxin. Those genes are found in the virus. And the virus is infecting those bacteria and making those bacteria pathogenic. So here's the question. So the bacteria, they want to infect animals like us and reproduce inside of us so they can make more copies and spread. But those pathogens are only pathogenic because they're infected with a dormant prophage. And the prophage, a bacterial virus, is carrying a gene to make a toxin that makes animals sick. But the virus infects bacteria, not animals. So why would a bacterial virus carry a toxin gene to make animals sick? I mean, it sounds kind of strange, doesn't it? A bacterial virus carrying a toxin gene that makes an animal sick, but the bacterial virus doesn't infect animals, it infects bacteria.
You're saying, like, why is there a middleman of the bacteria?
I'm saying, why is it that this bacterial virus would carry a toxin gene to make animals sick? I mean, it sounds kind of strange, doesn't it?
Yeah, maybe the bacteria can easily get into humans through food or through dirty water or something like that.
Yeah, but why do they need to produce a toxin? We haven't learned that yet, but we will.
Aren't they just producing what the virus is giving them? Isn't the bacteria?
Right, but why would the virus give the bacteria the ability to make a toxin to make animals sick?
Oh, I see.
Like I said, we haven't actually discussed this yet because we haven't gotten to disease. But when it comes to bacterial pathogens, it's the toxins that they produce that cause the symptoms of a disease. And these toxins, because they're making the animal sick, help those pathogens to survive and spread within the animal. Because if you take in these bacteria, your immune system is going to attack and destroy them, right? And so the bacteria need a way to help to stop that or slow it down, and they produce these toxins. So the bacteria want to reproduce inside of an animal host, but the animal host is going to kill them. And so the virus wants its bacterial host to be able to reproduce, because every bacterial cell is going to have a prophage in it. So the virus wants to reproduce. And so the virus is enabling its bacterial host to survive and spread within an animal host by giving it a toxin gene to make those animals sick, right, to make them weak. And that allows the pathogen to reproduce, and then it can spread to other hosts. And so the virus is doing this for survival. It wants to make more copies of itself. So the virus is helping its bacterial host to infect and reproduce inside of the bacterium's animal host. So, but isn't that fascinating? How a bacterial virus wants to reproduce itself, and so it helps its bacterial host to be a pathogen. So the bacterial host can reproduce itself inside of the animal host. That really is amazing when you think about, how awesome nature and evolution is, right? I mean, imagine a bacterial virus is carrying a gene to make an animal sick. And that gene allows its bacterial host to survive and reproduce inside the animal host, because now the animal host is getting sick. So it's pretty amazing when you think about it, right?
Yeah, I don't understand still why they don't classify viruses as something that's not alive. Like, it seems like it's...
Well, because right now the current definition of life requires that life be at least a single cell. So it's got to have cytoplasm, organelles, has to be able to make ATP. And so viruses cannot do that, right? They're parasites, forcing a host to reproduce it. So, but it is pretty amazing what viruses can do. And they can do an awful lot with a very small number of genes, as we'll talk about later on. But an amazing example of nature and evolution, right? So remember, evolution is about survival. And the viruses want to survive. And in this case, they're helping their bacterial host to survive. So now let's go back and revisit transduction. Remember, we said that there were 3 mechanisms of horizontal gene transfer. And transduction was mediated by a virus. So what were the other 2 mechanisms of horizontal gene transfer?
Transformation.
Yep. And?
Conjugation?
Right, so transformation and conjugation. So remember, transformation, bacteria had the natural ability to pick up short linear pieces of DNA from the environment. In the laboratory, we can force them, we can make them competent to absorb plasmids. And then conjugation involved those HFR and F plus cells able to make a pilus and conjugate with an F minus cell. And the F plus cell sends a copy of the fertility plasmid over to the F minus cell, making the F minus F plus. And the HFR sends a copy of some bacterial DNA over. And so the F minus cell stays F minus, but it may have acquired some new characteristics. So with transduction, bacterial viruses are the ones that are transferring genes from one bacterial cell to another. So let's focus first on generalized transduction, right? So generalized transduction involves lytic phages. And so remember, when a lytic phage first infects, the bacterial chromosome gets degraded. It gets broken down into little pieces. And when the new viruses are being formed, most of the time, the viral DNA is packaged inside the capsid. And so that new virus goes and infects other host cells. But sometimes, just due to random chance, a piece of bacterial chromosome that's floating around is incorporated into the capsid. So now that new virus is actually carrying just bacterial DNA from that host. So when that new virus particle, that new virion, actually goes and quote unquote infects another bacterial cell, the virus injects bacterial DNA from the previous host, not viral DNA. And so that very lucky bacterial cell is not actually being infected. It's receiving bacterial DNA, which may contain a beneficial gene, which allows that bacterial cell to survive. So it's a completely random process. Random pieces of bacterial chromosome get incorporated into the capsid, and those viruses may infect another cell, but they're not actually infecting them. They're injecting bacterial DNA, not viral DNA. So lucky for that new host cell, it's not actually being infected. And it may have acquired a beneficial gene. So that's generalized transduction involving lytic phages. Specialized transduction involves lysogenic phages. And so remember, the lysogenic phage incorporates itself into the bacterial chromosome, forming a prophage. So in this case, when the virus excises, when it cuts itself out, it actually cuts out a little piece of bacterial DNA next to it. And so this piece of bacterial DNA may have a beneficial gene on it. If it is a beneficial gene, then when these new viruses, right, carrying that beneficial gene along with the viral DNA, infects another host cell, the prophage that forms has with it an extra gene, which came from the previous bacterial host. If that extra gene is beneficial, this host cell is now going to survive better. It's going to reproduce more, which means the prophage is going to survive and reproduce more. So again, the viruses are trying to help themselves to survive and reproduce, but in the process, they're giving their hosts an advantage, which can allow them to survive. And this may represent what happened once and allow the lambda-prophage to pick up those nasty toxin genes. But this is a process whereby DNA adjacent to, next to the prophage, gets cut out along with the prophage. And so those new viruses that are created, they're carrying that extra gene, which may be beneficial to the next host they infect. So we call that specialized transduction. Again, these are ways to increase diversity in bacterial cells, allowing them to acquire a new characteristic in the same generation. So they don't have to evolve it, they can simply acquire it. And this process is mediated by bacterial viruses. So any questions on transduction? So we've talked about viruses. Remember, viruses are pieces of nucleic acid, either DNA or RNA, surrounded by a protein coat. And in the case of certain animal viruses, there will also be a lipid membrane that surrounds them. But it's basically just a piece of nucleic acid surrounded by protein. And of course, the viruses are infectious, they are transmissible, and they cause diseases. So there is something that's less than a virus. And that something is a viroid. So a viroid is simply a piece of RNA, and that's it. No protein associated with it, just a little piece of naked RNA. So sometimes these are referred to as subviral pathogens because they're less than a virus. They're just a little piece of RNA, no protein, and they're short, usually between 300 to 400 base pairs long. And single-stranded RNA is not stable, so it will often fold on itself and base pair with itself to provide some stability. Remember, that's why DNA is a double-stranded molecule, because being double-stranded and base pairing with another strand stabilizes the DNA so it'll last. RNAs are not meant to last, which is why they're single-stranded. But these are just little pieces of RNA that can be transmitted and cause disease. Now, so far, they've only been found in plants, so there's only some plant pathogens that are viroids. But if they exist for plants, they probably exist for animals, right? We just haven't found them. One example of a plant disease is potato spindle tuber disease. And so the idea here is that insects might be chewing on a diseased plant, and then they fly off or crawl over to another plant and start chewing on it and transfer the viroid that way. But it's just a little piece of RNA, and it's infectious and transmissible and causes disease. But it's just a little piece of RNA. So is there something that's less than RNA? What gets made from RNA?
Proteins?
Right, protein. So can proteins be infectious and cause disease? Obviously, since I'm asking that question, it must be true, right?
Yeah.
So there is such an infectious protein, and they're called prions, or as the Americans say, prions. But prion is the original pronunciation, so prions. And these are infectious proteins. So a protein that can cause a disease and be transmitted to others. And these are naturally occurring proteins in the brain, but you could have a genetic mutation where your proteins become abnormal prions. And then in that case, it's inherited, right? It's heritable. Or if you eat an infected animal, you could get infected that way. Organ transplantation with infected tissue, even contaminated surgical instruments. And the disease caused by prions are spongiform encephalopathies. I know, big words here. Spongiform encephalopathies. So spongiform means like a sponge, and a sponge is full of holes. Encephalo refers to the brain, and apathy is a disease. And so in people with spongiform encephalopathies, their brains are full of holes, and they look like sponges. We find spongiform encephalopathies in a wide variety of animals. In sheep, we call it scrapie. Humans have a variety of manifestations like Creutzfeldt-Jakob disease, Gershman-Straussler-Scheinker syndrome, fatal familial insomnia. So an inherited condition, fatal familial insomnia. So these people do not have the ability to dream. And we all dream. If you cannot remember your dreams, it's because you sleep so soundly that you just don't remember them. You have to either wake up or be in a light sleep to remember your dreams. But all animals dream. And it's essential for the brain to dream every day or every night. That's how the brain sort of processes what happened during the day and gets everything organized and gets refreshed, is while you're dreaming. If you cannot dream, you will die. And so these people typically don't live much beyond their teens or early 20s, and it's not a happy existence while they have it, because you can take drugs to make you unconscious, but that doesn't let your brain dream. So, but it's a type of spongiform encephalopathy where people cannot dream. But most people have heard of mad cow disease, the most common example of a spongiform encephalopathy, right? The cows just kind of start acting weird and do strange things. And the same thing is true of people that get these diseases. And so what actually happens? Well, as I said, you have normal prion proteins in your brains, right? They're perfectly normal. What happens is that if you have an abnormal prion, it has an abnormal shape. And that abnormally shaped prion actually causes normal prions to become abnormally shaped. So these are progressive diseases that usually take a long time to develop. And in humans in particular, it may take 10 or 20 years before you start manifesting the symptoms. And nothing can be done. There's no way to detect it until you have symptoms and you have a lot of damage. There's no treatment, there's no cure, there's nothing you can do. It's a slowly progressive disease where the normal shaped prions start becoming abnormal, and then it starts spreading throughout your brain. And when it comes to infected animals like cattle, the problem is that prion proteins, the abnormally shaped proteins, are unusually stable. They do not denature with heat. So cooking does not denature them. A well-done hamburger is not safe because prion proteins literally have to be burned to ashes in order to destroy them. They're unusually stable. So, cooking everything thoroughly does nothing to protect you from prions. So what is supposed to happen in meat processing is that any animal that's sick or behaving strangely is not supposed to be slaughtered and used for human food. And that's mostly true. Although if you search YouTube, you'll find lots of undercover videos where people went into slaughterhouses and things like that. And there are examples of sick animals being dragged to the slaughterhouse to be slaughtered. And those animals are supposed to be, thrown away, not used at all. Now, it's been a long while now, maybe close to 30 years, but in England had a problem with a massive outbreak of mad cow disease in humans. And so massive is a relative term just because it's rare. And so what happened is the farmers were trying to be efficient. And so the sheep farmers, most people, if they eat, they just like to eat the muscle, right? Most people just like to eat the muscles. Everything else is just, trash. But the sheep farmers are like, we've got all these extra sheep parts, right? All the internal organs, the brain, spinal cord, all this stuff. It seems wasteful to just throw it all away, right? And so the farmers got together and said, okay, well, let's be efficient about this. We'll take the leftover parts of the sheep that humans don't like to eat, including the brains, and we'll grind them up and we'll mix them in with cattle feed. And we'll feed the sheep to the cattle. And so some of the sheep had scrapie. And so a lot of the cattle got infected with mad cow disease, and then people ate the mad cow. And when this happened, our government assured us nothing like that happens here in the US, which may or may not have been entirely accurate. But the point is, they were doing something very abnormal. Because what are cattle supposed to eat?
Grass.
Grass, right? They eat grass, plants. But even in the US, most beef is not raised on grass because it would take about 5 or 6 years for that baby calf to grow to an adult, to be slaughtered. And so the farmers don't want to wait that long. And so what they feed cattle here in the US is corn. Corn is not grass. Corn is full of starch and sugars. That is not a natural food for cattle to eat. And so it does cause them to grow very quickly. So in about 15 months, that baby calf is full grown and is ready to be slaughtered, which is kind of a good thing because in another few months, it's going to die anyway because it's being poisoned by the corn. It's not a natural food for the cattle to eat. It's actually toxic to them. And so they are not going to live much longer than 15 months anyway before they die, right? Because corn is toxic. All those carbohydrates actually acidify the guts of the cattle. And that's not a normal environment. It's not supposed to be acidic. And the other problem is that by causing the acidic gut environment, cattle do have pathogens that people can get infected with. And so if you happen to eat meat, right, that is contaminated with some of these, bacteria, and you don't cook your hamburgers well done, your stomach acid will not kill these bacteria because they're intolerant now, because they've been growing in an acidic environment for 15 months. And so the way that cattle are raised is abnormal. And so when you do abnormal things, right, to nature, nature bites back. And so, that's my speech about meat. So what do you guys have for lunch? A nice juicy hamburger?
So when they say grass-fed beef, that means they were not fed corn, right?
Right, or at least mostly grass. But corn-fed beef has a mild flavor, and so that's why people like it.
Is that the same for chickens?
No, chickens, they can eat, seeds and corn and all kinds of stuff. So, yeah, chickens will eat a lot of different things.
Cool.
But anyway, so, the way that animals are raised is problematic because it can lead to problems, especially since we pump a lot of the chickens and the cattle full of antibiotics to keep them healthy because they're living in very harsh, conditions which are not, favorable to their good health. And so we give them lots of antibiotics. And so those can also enter the food chain. And they can also create antibiotic-resistant pathogens. So there's a lot of problems with the way animals are raised. So, any other questions? So, one last question. What kinds of things can be infectious? So, something to keep you up tonight. So, we already know that the most complex things are eukaryotes. And there are eukaryotic pathogens like Plasmodium and Trypanosoma. A little bit simpler are prokaryotes. And of course, there's many, many, many different bacterial pathogens. We've talked about some of them, and we'll talk about some more throughout the rest of the course. But those are cellular organisms. We now know that there are acellular organisms, right? Viruses, not cells, right? They're infectious, they cause disease. We just learned there's something less virus, a viroid, a little piece of naked RNA that can cause disease. And we also just learned that there's an infectious protein, right? Prion proteins that can cause disease. So when you look at the complexity of infectious agents, we go all the way from cellular with, a nucleus to without a nucleus to no cells at all, right? To just a little piece of RNA and then to a protein. So there's many different kinds of things that can get you out there. So this is something for you to think about. Are we worried?
A little bit.
Anyway, so, all right, so we're done with lecture, so we'll start lab around 10 after 1. All right?